Okay, great. I'm going to go ahead and share my screen. Is everyone able to see my screen?
Yes.
Okay, excellent. Go ahead, go in presentation mode here. So I guess first, hi everyone. My name's Amanda Saravia Butler. I am currently the GeneLab Science Lead. So GeneLab is part of the Open Science Data Repository project that produced a lot of the data that you guys got to work with this summer and over the TOPST course. And so thank you, James, for inviting me to speak to everyone here. My presentation is probably going to be a little bit different than the other two speakers because I just want to give you an overview of the Open Science Data Repository and what kind of stuff we have available for all of you guys. Some of you may already be familiar with this from what you've learned throughout the course, because I personally don't do my own research. I actually help facilitate the research of others through my role on GeneLab. So I'll start with an NASA Open Science overview. First, the definition of how we define open science. We define it as a collaborative culture enabled by technology that empowers the open sharing of data, information, and knowledge within the scientific community and the wider public to accelerate scientific research and understanding. So this is NASA's overall definition of open science. And as I'm sure you guys have probably learned in the course, space travel introduces a lot of different risks for living systems. We have categorized these risks into these 5 different categories here as distance away from Earth and confinement, so being in small spaces, making fitness difficult, as well as nutrition and the ability to sleep. And of course, that introduces stressors. And then those similar risks are also seen in the context of these being hostile and closed environments. And then, of course, different gravity fields poses a risk to living systems, as well as radiation exposure. And so because of these risks, we've developed different experiments in space biology research to help us understand how these risks affect terrestrial biology, to enable us ultimately to be able to live one day in space or maybe even on other planets. And so part of being able to assess this risk and understand it is to generate data from these experiments and make sure that those data are open and available to the scientific community to interrogate. So that's primarily what the Open Science Data Repository is for. It consists of 3 different projects here. So on the top, what you're seeing is the Ames Life Science Data Archive. And so this hosts data within the Open Science Data Repository involving physiological, phenotypic, imaging, environmental telemetry data, basically all sorts of data that are not omics are collected from spaceflight and analog studies. And then all omics data from spaceflight and analog studies are incorporated as part of the GeneLab project, also hosted on the Open Science Data Repository. And this can include data from a variety of different omics types, whether it be at the DNA, the RNA, the protein, or the metabolites level. And then some of the data that's generated in ALSDA and GeneLab derives from NBIS, which is the National Biological Institutional Scientific Collection. And so NBIS actually is a tissue repository that collects tissues from rodent experiments and other experiments that was brought to the space station, as well as shuttle missions back before the space station. And these are essentially all the tissues that weren't used by the primary investigators. So that way we can maximize, since every part of an organism was exposed to the space environment. So if the primary PI was just interested in the muscle, they would want to use the muscle from the animals in their experiment, but all the organs were also exposed. So in order to make sure that those are available to the community as well, NBIS collects those tissues that were dissected by another project called the Biospecimen Sharing Program. And then those tissues are made publicly available for anyone in the world to request. And so these projects altogether make up the Open Science Data Repository. And although GeneLab does generate some data in-house that is submitted and uploaded to the Open Science Data Repository, the majority of the data on there is actually from other researchers that have done spaceflight experiments and have submitted their data to our repository to host. And also we federate data from other databases. So if other researchers publish data in another database that was related to spaceflight, we'll actually find it and ingest it into our database to make sure that it is available, consistent within our metadata structure, which I'll get into. And so a little bit about what the Open Science Data Repository contains. So as I mentioned, it contains a variety of different omics data types. This is a breakdown of some of those major classes of omics. You can see that the majority of the data that we have currently is transcriptomics profiling. So this is measuring RNA or gene expression. And then we also have a variety now of physiophenotypic on the ALSDA side data sets. And so you can see a much larger mix here of different study types. And then in addition to having US-based studies and NASA-based studies, we also host data from international experiments. So that's just showing the breakdown of different international studies that we have in our repository. And then we do collect these data from any model organism that was in space. And so you'll see that the majority of the organisms in our repository is rodents, but we also have a fair amount of microbes and plants. And then for our human data, we have it broken down here into human cell lines, which a lot of work is done on. And then recently we got a huge surge of civilian astronaut data from the SpaceX Inspiration 4 mission, which is pretty exciting. And then we also have actually some JAXA astronaut data. Importantly, we do not currently host any NASA astronaut data. And then one of the most exciting things about OSDR is that because of the standards that we put forth within our repository and the accessibility in the community that we've built around this, these data are used to generate new publications or new knowledge about what's going on in the space environment. So here is just a plot that is showing in blue. The blue bars here are the publications from the original studies that produced the data that were hosted on the repository over time. And then the red bars are actually the publications that were produced by just reusing the data on the repository. And so as you can see, that number has been catching up to the same number, close to the same number of studies in the original publication. And we call that our return on investment. How much new knowledge can we generate without having to actually put up the funds to run a new experiment, which is very costly and expensive. And so, as I mentioned previously, anyone could submit data as long as it's from a space flight or analog mission to our repository. And so we have a submission portal that enables this. It's our biological data management environment, or BDME, because everything in NASA is an acronym. And so in BDME, anyone can create an account in BDME. And then once you create an account, you land on this homepage and you can create a new study to import your data. And then that will allow you to import the data associated at the sample level, as well as a particular assay that you use to generate the data. And each study can have multiple assays, so long as it was related to the same sample and often tissue as well. And then importantly, especially for the work that you guys have been doing with machine learning, is that all of the terminology for the metadata that's inputted for a particular study is connected to very controlled ontology. So that a spaceflight study is always called a spaceflight. The condition of being in spaceflight is called spaceflight. It's not called microgravity or some other term. So we make sure that everything is consistent, and this allows for more reproducibility and findability across the data set, as well as enabling things like machine learning. And this template is used both for omics data on the GeneLab side, as well as non-omics data on the ALSDA side. And then once a submitter uploads their data, they are presented with this view of what their study would look like once it's going to be published on the repository. But we actually don't publish anything until our principal investigators give us the green light to do so, because we want to make sure that they're able to publish their data first before we make it publicly available. And then once it's on the data repository, we have this search landing page here, and you'll see boxed in red is a bunch of different search features. So very similar to things that you guys are probably used to. With a search bar, we could do a keyword search to search for a specific data set, or you can use these organized search filters to search for either GeneLab or ALSDA data. You can also search for data on other databases that isn't ours, like NIH Geo. And I believe you guys might have used some NIH Geo data sets in your work. You can also search a particular data type, so study, or maybe you want to know more about the experiment or the biospecimen or the subject. You can also do search filters to search on the project type, so a spaceflight or ground-based analog study. There's also assay type searches and organism filters that you could use to narrow down the studies of interest. Those results are going to be displayed in the top right corner of this search. And then a list of studies will be displayed that matches your search criteria. And then once you go ahead and decide which data set you want to look at, you'll be brought to a study page that looks similar to this, that has a variety of different aspects of that study where you can learn more about it. So starting with the study description, to know what the context of that description is, it's very similar to an abstract. And then also at the top here, you'll have the OSD identifier, which is going to be the primary identifier for a study. And then it can also have up to 2 additional identifiers. So if a data set has GeneLab data or omics data, it'll have a GeneLab ID. If a data set has ALSDA data, it'll have an ALSDA ID. And then also all of the OSD studies have a unique digital identifier, digital object identifier here, a DOI. And then if you want to, you can click on this button to cite the study. So when people are using the data from this study in their publications, we make it very easy to be able to cite the data from that study. And then you can navigate through here through the different items related to this study. So I'll just show you one of those things. So you can navigate to the samples portion, and that'll bring up this samples table. And you'll see a table listed here where each row is a different sample for that particular study, and then all the metadata associated with that sample. So which organism it is, a specific strain, where that animal was purchased from, and then that goes all the way through to different factors that were applied to that organism, the sex of the organism, et cetera. And then all of this metadata, which again is standardized and uses controlled terminology, so it can be consistent with similar metadata features from other studies. You can pick and choose which information that you want and then export it as a CSV file. So this looks very similar for assay tables as well. So for each assay, you can select the assay that you want, and then you have a bunch of metadata with controlled terminology that describes that particular assay for each sample, and you can also export that information. Now, you can also go to the Files tab on the left-hand side for a study, and that's where you can access all of the files associated with that study. And so this can be cumbersome if you're downloading a bunch of different files. So we also have an API as well as a public AWS S3 bucket where you can more programmatically download data files that you want. If you notice here, this has GeneLab processed data files. So because omics data can be difficult to understand if you're not a bioinformatician, GeneLab works with the scientific community through our AWGs, which I'll talk about in a minute, to build these consensus pipelines. So essentially in the context of, for example, RNA sequencing data, raw data looks like this. Just a series of different sequences of A, T, Cs, and Gs, very difficult to interpret or comprehend unless you're a bioinformatician that has the tools to do so. And so we will take all of the raw data that's hosted on the Open Science Data Repository, and we will put it through our consensus pipeline to generate data products that are much more easy to interpret by more people. So something like what's shown here is a differential gene expression table. So very simply, gene names, symbols, what their expression values are in your spaceflight group, your ground control group, log fold change, and your significance values. And so again, this makes it really easy. And because all these outputs are standardized, we can do a lot of things with it, including using it for machine learning algorithms. So RNA-seq isn't the only pipeline we have. Our goal is to have pipelines for all the omics data that we host. We're not quite there yet, but we do have several other pipelines. Our methylation sequencing pipeline, our metagenomics pipeline are shown here on the right. And then all of our pipelines are publicly available on our GitHub repository, which you can access through this QR code, through this link. And a lot of these pipelines are also wrapped into workflows, making them really easy to install on a compute system and run for either OSDR data or actually any data set of the same omics data type. And because all of these standardized output products are, well, because all these output products are standardized, it makes it easy to implement things like this data visualization portal that actually reads in those standardized process data output files and generates these nice visualizations where you can actually get a visual picture of what's happening with the data, how closely related the samples are, and do different types of downstream analyses with these data without really having any bioinformatics expertise. You're just clicking and choosing from drop-down menus. And you can also combine samples from different data sets. In addition to visualizing the data products, you can also take a look at what the environment was like for a particular payload. So these are for rodent research missions. So if you saw something weird, let's say in your gene expression data and wondering, oh, what happened, you can go and take a look at the environmental data for that particular rodent mission and take a look to see if there was some anomaly in the temperature or the humidity or the carbon dioxide levels. And then also, if you want to compare different missions, you're able to do and look at the different environmental telemetry data from the space station for these different rodent research missions through the environmental data app. And you can also take a look at the radiation exposure from different dosimeters in different regions of the ISS and other spacecrafts as well through the RAD Lab data visualization portal. I'm getting close to time for my talk portion, so I'm going to fly through these. So this basically is just putting everything together. We're having this principal investigation and then utilizing the tissues that are not used in the principal investigation, making them publicly available through the tissue bank of NBIS. And then a PI can come and request that tissue, generate data from that tissue, publish that data in the Open Science Data Repository, which can then be reused to generate new knowledge. So this is just a QR code for a paper that was published that uses QLattice explainable AI, utilizing data from the different data sets hosted on OSDR to identify relationships between calcium aberrations and molecular biomarkers in spaceflow muscle. And then I mentioned previously, I alluded to working with our scientific community. So our scientific community are referred to as the analysis working groups, and they're broken down into different specialties. So you see animal, multiomics, microbial, plants, AI/ML, and also ALSDA. And what these groups do is they give us feedback on our ingress and egress standards. So how we ingest data and then how we display data on our repository to help make sure that these data sets are standardized, consistent with specific subject matters, and also are easy to find and reuse. And then ultimately they work together to generate new knowledge, and they publish on those knowledge. So here's just some different QR codes pointing to different publications that were generated. So furthermore, to make the OSDR data accessible to more people, we have developed training programs using OSDR data. So these I'm going to go through real quick. The Gene Lab for Colleges and Universities teaches bioinformatics at the college level. We also teach both students and educators. And so we do this by organizing information into introduction modules and then omics specific modules. So that way you don't have to come in with any coding experience. We give you an introduction to coding and then walk you through a particular omics type. So this is looking at two different, we do this using Jupyter notebooks. And this is just looking at what our Amplicon-seq and RNA-seq Jupyter notebooks look like. And then, as I mentioned, in this approach, we teach both students as well as educators. When we teach educators, we provide them with all the resources to teach in their home institutions. And then people who complete it will receive a certification for the particular omics type that they completed. And I'm currently working to put this on demand. Hopefully it'll be ready by this fall. And so then you guys will all be able to access it. And it's through Canvas, so the same platform that you guys are all used to. This is just showing some metrics of the effectiveness of the boot camps that we've hosted so far. And then we also have a version of this for high schoolers. This is called Gene Lab for High School. And these are for rising high school juniors and seniors in the US. And they spend 4 weeks learning about bioinformatics and RNA-seq, and then they process a data set and come up with a hypothesis. And then make a proposal based on their hypothesis and present it to folks at NASA. And we also have a teacher version of this program as well, where the teachers take the Gene Lab for High School content and make it their own, so that way they're able to teach it at their home institution as well. So this is just describing the course content that I had summarized. And then you'll see that we have had students from a variety of different states throughout the US, and then just looking at some metrics breakdowns of Title I versus non-Title I schools, as well as male and females in this programs. And then I don't know if James talked about the TOPS 101 training, but this is the general open science training program. And so if you guys haven't taken TOPS 101 yet, I highly recommend it. It introduces you to open science, the concepts around open science and data management. And then this, I'm not going to go into, this is the course that you guys all took. So congratulations on being the inaugural class for the first TOPST AI ML training. And then, as you probably already know, James offers this as well asynchronously through Canvas. So you can take it on your own time if you guys want to go through it all again for a refresher later. And then I just have this slide here that shows additional opportunities as you guys get further along in your careers for different internship programs that are offered, postdoc programs, as well as pathway programs to open up a spot at NASA and then STAR when you get a little bit further along. And then I work with an awesome team of people on GLab, ALSA, and NBIS, all of the Open Science Data Repository. And so big thanks to them and all of our support. And I think I used my full 20 minutes, but if you have any questions, I'm happy to answer them.
Awesome. Thank you, Amanda. I actually do have a quick question, and if anyone else has a question to follow up, please either write it in the chat or unmute yourself and ask. But my question, there were actually two. One is you mentioned that I know we don't share astronaut data. Do you see that ever changing? Like, especially given now we have the Inspiration 4 kind of data out there, do you think that might kind of lead the way to open up data for astronauts or not necessarily?
So for NASA astronauts specifically, that's a big challenge just because of all the red tape and how tightly protected NASA astronauts are through the human research program. So I don't foresee that happening anytime soon. However, for commercial astronauts, we actually have more and more collaborations with upcoming commercial astronauts flights that are planning to submit their data. And as I mentioned, this is just another presentation where I believe on here somewhere I have an example of what you're looking for. So for the I-4 data, the Inspiration 4 data, for example, that was from this mission from 4 different astronauts, first ever commercial flight mission. They took a bunch of samples, both pre-flight, during flight, and post-flight. And so these have all been organized in different studies on the repository. And for a lot of these studies, all the process data, so the non-identifiable data, are publicly available. However, the sequence data from the commercial crew themselves are of course identifiable. And so those we actually do have available, but it's through a controlled access request process. So if you do need access to the raw data for whatever reason, there is a little button to request access to those restricted files. And then it goes through this review board process. And then you are given access at the end of that following a particular set of rules. So yes, available in different forms.
Got it. So the FASTQ is the raw data that you don't share, but the summarized, the processed files like the count data, you do share without the restriction.
Exactly.
Right?
And maybe not the individual count data, we're still discussing that actually, but like the combined differential expressions. So like the data from comparing two different groups. And so we're still talking about and actually working with the IRB from the I-4 to see if we would be able to provide count data publicly. But any of the data is available through controlled access. So you just have to go through this controlled access request and justify your reason and what you're doing with those raw data or any of the higher levels of process data, or lower levels, I should say, of process data that might be behind that controlled access.
Got it. Cool. Thank you. Thank you very much, Amanda, for sharing that. Does anyone else on the call have any questions for Amanda? We can also wait till the very end. There's someone who does. Matthew, go ahead.
Yeah, I had a question. So like I'm a high school like rising senior right now, and I'm interested in going into like space biology, especially like the bioengineering component of it. What would you like recommend as my like next step as I
So rising senior right now, meaning you're going into your senior year of high school?
Yes, next year I'll be in my senior year or this school
Okay.
year.
So unfortunately, that means we won't be able to enroll in the GLab for high schools because it's they actually just finished recently this year's cohort of GLab for high school students, and next summer you will no longer be a rising junior student, you will have graduated. However, what I really highly recommend is joining one of our AWGs. And so depending on if you're interested in a particular organism or if you just want to do more AI ML stuff, you can join the AI ML AWG. This is going to give you access to the people in here are at all different levels. So you'll get folks in high school like you are all the way up through very established researchers and PIs, both in academia and in the private sector. And it's great because it's a great opportunity for networking, to show off your skills, to work on a project with other scientists with similar interests. And then anytime we have opportunities available, the first place we post those opportunities are to our AWG distribution list. And so I definitely highly recommend that. And then once you're into college, I'll give James the information for Gene Lab for colleges and universities once I finish the on-demand course. So you could take that if you want to learn more bioinformatics on your own. And then as you're in graduate school, there's a bunch of different programs that you can apply to at NASA. So there's the NSTGRO, which I don't think I had on the slide. You have an intern in that right now. And the SLSTP program, which I do have on here, which is an internship program that you can do at NASA. And so there's a bunch of these opportunities for you to apply to. So I'm happy to share this slide deck so that way James can pass it on to all of you guys. And then if you have any questions, you're also more than welcome to shoot me an email.
Excellent. Thank you. And I shared a lot of these opportunities just so everyone knows. A lot of these were posted in the discussion on that same topic, like what do we do after this class as follow-ons? And I think most of this, if not all of this on this slide and Gene Lab for university, Gene Lab for high school is also listed in that thing, in that discussion on the Canvas system. So make sure to check that out. Okay. Thank you very much, Amanda. Again, I appreciate
Just
that. Sure.
one more recommendation.
Yeah.
Take statistics courses. Take as many statistics courses as you can in biostatistics while you're in school. Statistics is one of the most difficult things to learn on your own. So while you're in school, take stats courses. You will need it in any discipline that you enter into. So I can't stress that enough. That's one thing that I really wish I did more of.
Yeah,
Okay,
I would
now we can
definitely, I would definitely agree with that. And for me, where my lacking was is in biology. So I had the computer stuff and the statistics and the math, but not the biology. So I would also recommend taking biology when you can. Awesome. Okay. And so our next speaker is Marshall. Marshall, go ahead. And you may be muted. I'm not sure. I think you are muted, Marshall.
Okay, I'm not. Okay, good.
Yep.
So, is that good? Okay.
Yes, we can see your screen and we can hear you.
Okay, sounds good. Sounds good. Okay. Sounds good. Sounds good. Sounds good. Sounds good. Good. Okay. So, yeah. So here, basically, when astronauts go to outer space, they can become their biological age can increase. You know, biological aging is quite different from chronological aging that accumulates with time. And so they have shown signs, genetic from studying their genes, they've shown signs of frailty, which is a clinical condition that describes biological aging. And so here I try to put up, I try to develop a workflow that can classify samples for frailty. And of course, the purpose is what? What I'm using here is, I'm using here a data set. I'm using data sets of, very well measured samples collected from humans that are already known to be either frail or young. And I'm going to develop it from a very well classified database, which is NCBI. And so what I'm trying to do is to obtain, the purpose of classification is you want to come up with a number of classifier genes for that condition. It's usually a small set. And then the next thing you want to do is to prove that the set of classifier genes that you have is authentic. You're now going to do, or you're going to test now that classifier genes against those classifier genes against samples that are already very well known, samples whose class or category is already well known. Okay, so that if the majority, simply the majority of test samples are rightly classified, then you consider the workflow that determines the classifier genes is an authentic workflow. And then to add that usually there is something that is done in classification. Usually the training samples are usually the samples that you use to determine your classify your genes, and then you can have test samples, that is those that you will use to test. That is, you already know to what class those samples belong, and then you use them as a test to see whether your classifier genes or your algorithm will correctly detect them as, will correctly determine their belonging. And so that is the process of testing. But in some cases, when you have an entire data set, you will divide it into a training data set, which you will use for the training, and then you leave a few samples that you call validation samples. And the only reason you call them validation is because they are samples from the main data set that you use to obtain your training samples. So it's simply validation is simply that testing procedure that is within the same data set that you used for training. So that when you're done validating, you can go further to authenticate your algorithm, or rather authenticate your classifier genes by testing across data sets that are beyond the range of the data set that you used. Okay, so in this case, I'm using data which is muscle tissue that was extracted from frail, fit, and young males, phenotypically classified as frail, fit, and young. And that RNA-seq data for those muscle tissues is stored in NCBI GSA, NCBI, which is a very well-known database. And here, I'm interested in something particular. I want to collect all frail samples, whether they are male or female, and all young samples. And I want to just see what I can use that to train my algorithm. And so that I have a number of about 58,000 genes at the very onset made up of ensemble IDs. And of course, the things that you're going to have to do, you're going to have to transform that gene nomenclature uh, to gene symbols. So we're going to, first of all, we obtain the data and we do some processing. But first of all, let me describe the classification approach altogether before I talk about the data processing. So I have what is happening. I have about 24 samples that are frail and 26 that are young. And so but I end up using only 22 samples each from the frail and young samples, and I'm going to describe later on how and why. And then I use 15 samples each from each category as training samples, so that the remaining 7 will be used as validation samples. And then once I have that, I develop a workflow from the training data set to determine now my small set of uncorrelated genes. So my classifier genes, most important, all the genes have to be uncorrelated. And these genes will adequately classify any RNA, any sample having RNA-seq data as frail or young. So that at the end, I take a validation sample. If I pick a validation sample at random, I already know what its phenotypic class is. I'll simply test for its frailty by determining whether the closest training sample to it is frail or young. Okay. So I prepared a data set. I use R language, not Python. I do that in R Studio. And so the first thing I do is I take off any outlying genes. Outlying genes can be identified in DSeq2, which is a package in R, and I removed all the outlying genes, identified 51 of them. There is a well-established procedure in how you can identify outlier genes from their counts, and how their counts do not seem to be in rhythm with the rest of counts, across the samples. After taking off outlier genes, I then normalize all the genes. Okay, still in DSeq2 package, I normalize all the genes. And then one thing I'm going to try to do is I'm going to try to do some clustering of all the samples. I want to see those samples because the phenotypic class of those samples is very well defined. And I want to just do some clustering to see whether those samples truly belong to their clusters. And here I'm using something rather a little bit simpler or maybe primitive, multidimensional scaling. I can just do an MGS plot. I can see all the samples in 2 dimensions. And I can also use a heat map of the sample to sample distances. Okay. And this will be described later on. And then after I'm done with all that, those will enable me take off any samples that are outlined. And then after that, I will annotate the genes. As I said, I have gene IDs, I annotate them as gene symbols, and I use a package called Biomart. And when I do that, if there are any duplicate gene symbols, I remove them. During the annotation, there are also gene IDs, that is ensemble IDs that do not have a symbol match. Those are also taken off. I also take off any genes that do not have a significant total count across all the samples. So that when I'm done with all that, out of initially 58,000 and something, I'm remaining with about 20,851 genes. So as I said, the first thing is I want to see when I take all those samples, I take all the samples in my data set, it's 24 uh, 24 field samples and 26 field samples. I do an MDS plot. Now, what does an MDS plot do? It shows you 2 dimensions, simply the distances between the samples in the plot as you visualize them correlates with the dissimilarity between the samples. So if this distance is too big, it means that those samples are dissimilar. If they're too close, the samples are similar. When I look at it, first of all, as you can see, I have 2 samples that really look completely distant from the rest. Okay, and it does not enable me to even visualize the other samples, how they space. So what I'm trying to cleverly do is I have so many samples. I don't mind taking a couple of samples, and remaining still with a big number that can help me in the training and validation process. So that's what I'm going to do. I'm going to take off these 2 samples that are so distant. The next thing is when I'm remaining with that, now I do the MDS plot again for the remaining samples. And you can see it looks like you can clearly separate samples that are frill seem to be in one space, right? When you look at the upper left, and then samples that are young, when you look at the lower left, you have the samples that are young. Good. And so this kind of picture shows you that yes, the phenotypic classification into frail and young is somewhat justifiable when we look at the genetic basis. But nonetheless, when you look at the boundaries of the separation, right, when you try to draw something like a boundary between those 2 regions or space, you will notice that there are some samples from either category that seem to somewhat wander slightly into the other region. And so I when I look at them, I try to detect what those samples are. They seem to be about 5 of those samples, 4 or 5 of those samples. So I have about 48 samples. And so if I take off 4 or 5 of those samples that seem to somewhat mingle, a little bit, although maybe not too much, but somewhat significantly in the other group. If I take them off, I can determine those as outlying samples, and it will be good that I can have 2 distinct groups that lie in their own category. So that is what I do here. I remove about 4 samples that do not belong to their original category. And likewise, that is the MDS plot. If I show the heat map is going to show something similar. You are going to see that the heat map, have a heat map of sample to sample distance. And this heat map does some hierarchical clustering. All samples, if you see a sample that is mingling, if you look at the rows and the columns, and if you see a sample that finds itself in the category, not in its own category, that probably is a sample that looks like an outlier. For instance, you can look way down If you look at the right, and at the bottom, you see S514 female young, it's finding itself in between frail samples. And it's true, that was also confirmed by the MDS plot. That particular sample also finds itself in the opposite category. Okay, and so such samples, when I clearly identify them, I take them off. And so I end up taking 4 additional samples, 3 of them young and 1 of them frail, so that I end up with 22 samples in each category of frail or young. And when I show an MDS plot, you can see at least something reasonable, like a clear division between them. These are the samples that I'm going to use for both training and validation. And of course, the heat map can also show the clustering similar to what the MDS plot does with the 22 samples. Okay, now in a perfect scenario, like when I did it only with male samples, when you look at a heat map of samples, the sample distances, you will notice 2 separate clusters, right? One cluster is having only the frills, and the other cluster is having only the young. But in my actual scenario, where I've mixed both males and youngs as one frill, and as both males and youngs, and one in each category of young or frill, you can notice that although you can point out, you can pull out clusters of young and frill samples, subgroups, still there is some mixing. It seems to see there is some mixing in the heat map, and it's difficult to pull out a clear separation, but it's still fine. Either way, we're still going to see how this turns out to be when we're going to work out the algorithm. Okay. So the next thing, I have this big pool of 20,000 genes. I want to select the genes that are, first of all, relevant to frailty. Which are those genes that will be able to clearly distinguish, between, samples that are at a short distance from one another, which genes would clearly distinguish the 2 conditions, frail or young. And so when a gene can clearly distinguish, it is given a higher weight. And if it seems to very weakly distinguish, it's going to be given a lower weight, that is a negative weight. And so how do we do this? We simply, let's say you take a sample randomly from my data set, right, of all samples. And for that sample, what I'm going to do is I'm going to find the nearest neighbor samples to it, okay? Nearest neighbor samples of the same class, of the same phenotypic class, I'm going to find those. And I'm going to call them hits in the letter H. And then I'm going to find also what? I'm going to find nearest neighbor samples from the contrasting class called misses, in the uppercase M. So I'm going to do this randomly for 20 samples. And here, the number of nearest neighbors in each case is going to be 9. So this is how the algorithm works. Initially, all gene weights are set to 0. So I don't know how much each gene contributes to, determining to discriminating between, the different phenotypes. So now, how does a gene accumulate weight? Okay. Now, if the distance between, for that particular gene, if the distance between my sample of interest and its neighbor, right, which is of the same class, right? If the distance between them is less than the distance between that sample and a neighbor of the opposite class, then that adds a positive weight to the feature, because that is what I expect. Well, I'm expecting that for any particular gene, if I have 2 samples, the Euclidean distance is supposed to be smaller. That is to say, the genes are supposed to express themselves similarly if the 2 samples belong to one class. Now, if the 2 samples belong to 2 separate classes, I'm expecting that that gene should show dissimilar expression. That is to say, the Euclidean distance in gene expression between those samples should be bigger. And when the distances is bigger, you give it a negative weight. When the distance is smaller, you give it a positive weight. And that is how the genes across all the samples, across all their nearest neighbors are going to accumulate weights. So that is This algorithm belongs to the class of relief algorithms that are used to determine relevant genes. Okay, next thing is, once, let's say this is the result, my genes originally were 20,000, and I have survived when I use a weak criterion of if my weights, I use a positive weight 0.005, all the genes that have a weight above 0.005 are deemed relevant to frailty. I come up with 12,000. Now, if you're more strict on the weight, if you want the weight a little bit high, like 0.01 or 0.02, maybe you end up with about 6 or 8,000. It just determines on how many you want to end up. At the end of the day, you want to end up with a small number of uncorrelated genes. Good. Now, the next thing is, after you've obtained the genes with relief, you should be able to note that this algorithm determines relevant genes but does not detect redundancy in the gene function. That is to say, there are many genes that you're going to determine that are somewhat correlated in biology, or in other words, they belong to the same network. And so the next thing I want to do here is I want to develop or implement something like a correlation algorithm that is going to determine genes that are correlated with one another. When there is a correlation between genes, I'm going to have to choose one gene, so that I'm going to end up with only uncorrelated genes that will be my selected genes, and these genes will constitute my set of classifier genes. Okay. So how does the correlation work? I use an algorithm called a distance correlation. You know, all what I simply do is, yes, the distance correlation is something that measures a dependence between two random vectors. Okay, you have a mathematical formulation, a row of x, y equal to 0. That is the distance correlation is 0 if two vectors x and y are independent. If it is greater than 0.5, I'm choosing a threshold of 0.5. If that dependence is greater than 0.5, I'm going to say my two genes are dependent. If it is less, I'm going to say my two genes are uncorrelated. You can also here choose this correlation threshold. You know, you can choose a stronger restriction like 0.3 for your correlation. So when I do implement this, out of the 12,000 genes that were relevant, 94 genes survive. These 94 genes that survive, uncorrelated, become my classifier genes. Now, let me show you what I obtained. This is an Excel. This is obtained from this is the table obtained for, as I pulled it out from the code, it went onto an Excel sheet that I pasted here. These are the 20 classifier genes ranked by their weights. Yes, this is 20 classifier genes ranked by the weight. That is, you know, genes that will be able to clearly discriminate between the two phenotypes. And these were out of the 94, these are the 20 top classifier genes. So it goes from SCAP2 all the way to TWIST2, and you can see these genes that are somewhat familiar, most of them with frailty. And then the next thing is, after I have obtained my classifier genes, so this is all what I need, which means that with 94 genes instead of 20,000 or 50,000, which is computational load, these 94 genes, okay, can be able to discriminate, can determine whether a sample is frail or young. And so the next thing now is I prepare a validation data set. I have, as I said, I have 7 young and 7 frail samples with annotated genes from the original subset. And so my validation data set now is constituted from the select classifier genes. That is, only genes that are among the classifier genes make my data set. Okay. So now when I have now my validation data set, what do I simply do? For any given validation sample, I calculate now the Euclidean distance between the validation sample and a number of randomly selected training samples of each class, young or frail. In this case, I'm using all training samples. So I take a validation samples and I calculate the distance from all 15 training samples in each class, young and or frail. And then what I do is now I rank all the samples. That is a total of 30. I'm ranking all the samples by their Euclidean distance in ascending order, you know, for each validation sample, right? I rank all the training samples. Now, The validation sample, how do we give it a class, is assigned to the class of the training sample that ranks first, since it is of closest distance. That is a criterion of classification I'm using. And in this approach, when I've done the test, all the frill validation samples are classified as frill correctly. And for the young validation samples, 6 out of 7 are correctly classified as young. And so that the young validation sample that do not classify is S518. And so there is an output of tables of ranked training samples for each validation sample, as I can show you here. This is an example for my validation sample S301M and the male frill S301 here on the left. That is a test validation sample. I rank all the training samples by Euclidean distance. As you can see, the top, that is the shortest distance, is a frill sample, even if it's closely followed by a young sample. But you see that the remainder of samples are closely followed are frill, and then the young samples are down at the bottom. That is far away from it. And so that sample correctly ranks as a frill sample. And then when you look at the young here sample on the right, S529 frill young, you can see the same thing. Here, practically all the top sample is a young, and practically all the young samples, are closest to it, apart maybe from one sample, which is a little bit distant. And of course, all the samples that are distant from it are frill. Good. And so that is how the algorithm works, and it shows promising results in correctly testing any samples. The validation is very good. And so, yes, I think that this is something I'm wrapping up with. So you any further improvements, maybe besides just using MDS plots and the heat map clusters, I can develop like a clustering algorithm such as k-means. I would implement k-means just to confirm maybe what I have in MDS, or maybe my heat map as a good way of clustering the working data sets. And so please, that is the end discussion. The slides are open for discussion. Thanks.
Awesome. Thank you, Marshall. There are some questions in the chat. One is from Lanuki, who asks whether you'll be using ggplot in R to cluster the data. Is that the library that you used?
So let me explain to her. Well, ggplot is a package in R with many capabilities. And yes, let me see. ggplot Pro, I think MDS plot comes from Lima package. Let me see. The heat map that I produced, I don't think ggplot, is providing mostly graphics capabilities. So maybe the heat map I produced is using ggplot. Okay. But clustering does not really have anything to do with ggplot, the clustering process itself. Is that good enough, Linuki?
Lanuki, you might be on mute.
Okay.
Lanuki, did that answer your question?
Okay, she'll get back. I'll put it in the chat. I'll write it in the chat. But we can get to the others. I'll write this answer in the chat.
Okay.
Okay.
And there's another question, and that comes from Amanda. Do you have a heat map with only female samples?
Okay. Let Amanda. Yeah, well, let me see. Let me show you something. The first thing I did, yes, you're talking sense. I first did, like I said, I first did this for only male samples. What I first did, I used the male samples for training and then used the female for validation. So yes, if I put up a heat map of the female samples that I use for validation, you'll see the same thing as the male samples that I've shown here, because they were perfectly selected, like a true of all outliers that were close, very well selected. You have a heat map like this. But later on, because I want to be classifying frailty, but not with respect to gender, but frailty as a syndrome, later on, as what I've shown in this presentation, I mixed both male and female, in one group. Okay. Is that clear, Amanda?
Yeah, that's clear. And I was just curious, right? Because I wonder if you also have the metadata stating what stage of the estrus cycle that your female samples were in during sample collection, right? Because estrogen levels are going to change when you get older, they're going to be less, but then they also change during the female cycle. So I'm wondering if some of your outliers that were young but look like frailty might have just been because they were in the stage of their estrus cycles where the estrogen is lower.
Wonderful. Yes, Amanda, I very highly appreciate that discussion and that suggestion, because those are the kind of things I was having in my mind. When you do this, those who are phenotypically young, as you saw in my algorithm, I was happy to find out something. Frail samples classify perfectly as frail in most cases when I test my algorithm 7 of the 7. Now, the young samples, a few of them, like I said, 1 out of 7, right, you saw showed up to be frail. And so that is it. You can have some young people may still show signs of frailty in their genes. And especially for females, as you said, that could be the case sometimes based on their cycle. If the sample is taken based on the sample, you can have, uh, you can have a modification or an offset in the gene expression. And those are the kind of, so that's an important point that you're making here. Okay.
Yeah, and I'm also just curious, when you got your final list of ranked weighted genes that were determining frailty, that were distinguishing the two groups of frailty and young, did you do like a pathway analysis on those to kind of see what pathways are implicated in young and frail groups?
Well, that is fine. That is a thing to do next. You just right. So the question is, I may not really do something like a pathway analysis, or maybe you're saying for those 94 genes, yes, I have the data set. I can submit that in something like GSA and do a pathway analysis. But something I could also more easily do is I could do something like gene ontologies, maybe to see, yes, to see what pathways are involved from those. Yes, that is the next thing I'll probably have to do, to see what pathways, as you say, are probably associated with those genes, if they have to make any sense with frailty. But good stuff is they do classify somewhat adequately.
I'm also sorry, one more question. It's just really interesting, Mark. Did you have mitochondrial genes in your data set? And if so, were you able to pull any of those out? Because we know that mitochondria plays a big role in aging.
Okay, yeah, good. So your question is, can any of the genes that I have be identified as mitochondrial genes? Is that the question you have?
If they were in your original data set, yeah.
No, that's not something I have done. If you want, you can do it. For instance, it's open. That will be fine. I've not tried to identify, like, are they mitochondrial genes or are they simply nuclear genes or something like that. It's just genetic. It's just RNA-seq, general RNA-seq. Okay, good.
Okay,
And all of what you are saying makes sense. It could be interesting to see how mitochondria is playing a role in all this.
Yeah, thank you for that. It's a super interesting project.
Yes, but it's good to remind that these are not astronaut samples. These are regular, samples, people on ground, with those who've gone old, above 65, when they show the frail phenotype, muscle, based on how their muscle reacts, their gait, their walking speed, their gait and stuff like that. They are deemed frail people, and then there are the young people that we definitely know are not frail. They are the ones that provide the samples. But of course, the end goal is what? Once an algorithm like this is working quite well now, I can use it now to test astronaut samples, those that have flown to space and come back, right? That is it. I can now use it to test samples that have been to space, to classify are they frail or not. Okay.
Yeah, I'm super curious to see what those data turn out to be.
That's right. Okay. Good.
Awesome. Thank you, Marshall. There is one last question, and I think we can ask this question of all three researchers. I think the answer is yes, but I just want to make sure. Is it okay for the students to reach out to you directly with future questions? And if so, could you just drop your emails in the chat, and then those students who are interested in reaching out to you could. That would be great.
Yes, that's fine for me. I've dropped my email already.
Awesome. Thank you. Okay. Thank you very much, Marshall. And our last presenter is Dr. Richard Barker. Richard, do you want to share your screen and do a quick sound check?
Check 1 2 1 2. Okay, here we go. Let's see if I can share that screen.
Yep.
Okay.
We got you.
Let's click on the go to slideshow mode. Okay, cool. Okay, cool. So that was an awesome example of how you can develop machine learning algorithms using terrestrial data sets to help create new insights that can be applied for astronaut health using open source data that's now available through the Open Science Archive, such as the Inspiration 4 dataset. I'm going to talk about a astrobotany research program that I collaborated on in the Gilroy lab, which ended up using a stress pattern recognition system, a form of machine learning, to inform future proposals that led to cotton being sent to spaceflight. This is my cover slide. Essentially, I list all of the awesome people I collaborated with in the Gilroy lab. Too many to mention based on the time I have. It also lists some of the funders like NASA and NSF I need to thank, and the British Biotechnological Research Council for starting me on my research journey. The other thing it has, which is kind of novel in space biology, is mission patches. And so like TikTok, Toast, Blast Off, and Tasty, these are all mission patches associated with some of the projects that we've had the opportunity to fly because of the data analysis that we've performed. So hopefully there'll be some inspirational goals for you. Okay, but I want to show you my origin story too. I've got a funny accent. I come from the old world. I come from Norwich in England. Around my hometown, there's these wonderful yellow fields where the agricultural industry has got really efficient at growing mustard. And so they have a fantastic mustard shop. You can get Coleman's, and they made so much money, they sponsored my local football team. So these commercial enterprises had real social impact. What I learned from this system is that brassica, as in mustard plants, can become a really good canary in the coal mine and help provide new insights into space flight environments to help us create more sustainable growth moving forwards. What took me on this journey, one little thing from the past, like we're talking about different things that are really useful to practice. I recommend when you go to college practicing photography or biological photography. This was me practicing biological photography at college. What I learned was in order to capture the fire trajectory, I had to do a long exposure. And so it turned out this particular technique that I was practicing for performance art music festivals also came in useful in the laboratory. The data I produced gave me a chance to start playing with code and seeing how I would look at changes in the movement over time. And then when I went to the University of Wisconsin-Madison, I had the opportunity to use fluorescent microscopes, where essentially, again, you use long exposure to capture really faint fluorescent signals. In this particular video that you see, we have a GCAM fluorescent sensor. So it's a calcium sensor. And so when calcium goes into the apoplast, sorry, into the cytosol, this fluorescent protein changes in intensity. So when we wound the leaf and then add 100 millimole of glutamate, we can see this calcium wave propagate from the wound site to all the different tissues. I like to call it the Christmas tree effect because it kind of lights up like a Christmas tree. What this genetic tool allowed us to do was observe this system and how it responds to other stressors. So here we see that same technology being used to observe leaves as the root system gets flooded. So about 5 minutes after the root system floods, we can see that the vasculature lights up. So this is that calcium wave propagating, and it even causes these hydrothodes on the ends of leaves to start to glow. The moral of this story is image analysis is fun. This passion of mine stemmed from time-lapse photography, and there are lots of machine learning and artificial intelligence opportunities associated with automating these types of image analysis. But with this tool, we then realized we could start to probe some genetic systems. I really want to understand how this calcium system works. And so we started looking at some of the reviews that were being around. So essentially, plants have a long distance systemic electrical signaling system. It's not a nervous system, but it does have an electrical component, along with a calcium component, and along with a wave of reactive oxygen species that propagates along the along the apoplast outside the cell, sending the signal from one cell to the next. As a classical geneticist, one of the way I learn about things is by breaking them and then seeing how the system then responds. So essentially, what we were able to do was find mutations in these two genes, this ARBOD and this CAX2 gene, that essentially have a role in producing reactive oxygen species and a role in moving calcium ions and protons into the vacuole. These two systems are known to be associated with this long distance calcium wave. So here we can see we have these two mutants in this CAX gene, so two different alleles, and we can just see when we flood them, they have a larger rosettes and they don't get this purple pigment that forms in the wild type when it gets stressed. So ultimately, what I'm trying to say with this slide is this CAX2 mutant appeared to show a flooding resistance on the ground, and we were able to use that as preliminary data to propose that it might provide resistance to spaceflight, because the microgravity environment of spaceflight might create local environments of hypoxia due to lack of convective mixing of gases. And this could be particularly important for plant roots because they can't move in the same way that animals and leaves kind of can. So this was a really exciting day for me. I got to go to Kennedy Space Center. In the top left-hand corner, you can see myself and Dr. Swanson sowing seeds on Petri dishes. That's the Arabidopsis thaliana's natural environment, a square Petri dish. This was Dr. Simon Gilroy, Professor Gilroy, loading the samples. You can say that Teddy's a good professor. He's rolled up his sleeves and he's getting his hands dirty in the lab. Essentially, this fire-red light makes the seeds go into a deep state of dormancy. We can then wrap them in tinfoil and they won't germinate until they receive a light. We hand them over to the people at Kennedy Space Center, who put them into the SpaceX 13 rocket. This was the moment when the rocket started to take off. We were just the other side of the bay, just here on the other side of the Banana River. My boss, Dr. Swanson, had her phone in her hand. She was giving us the countdown, and then you could see the plume of smoke. I knocked the phone out of her hand. I was like, there's a delay on the internet. Look, it's taking off. It's taking off. She looked up, and it was really weird. Things weren't quite as you kind of imagine. Like, I've just been to the 4th of July. I know what fireworks like, but when the SpaceX Falcon takes off, it's slow and kind of ominous, and you can see the sound wave coming at you. People say you can't see sound. No, you can. Like, the water kind of splashes, like the mangroves shake. You feel the sound wave come up through the base of your heels, then the back of your ear, and then whoosh, the air and the sound hits the front of your face because, you know, the sound waves travels faster through the ground. Anyway, around this point, everyone starts acting weird. I saw these nice Japanese gentlemen like bowing to one another. These American people were giving each other high fives. I saw my bosses give each other a kiss. Yuck. And then there was this Italian guy. He was really quiet. And I turned to him and I was like, why aren't you jumping with excitement? And he was like, well, I'm waiting for the burn. And I went, the burn? What do you mean the burn? And he goes, look, the burn. And at that moment, like the engine kicked in again. And suddenly the Falcon 9 was flying across the sky. It looked just like Superman in the movies, but without the red and blue streaks. It was really surreal. It's like for the first time in human history, there was an intercontinental ballistic missile flying straight at us, and yet we weren't afraid because we knew it was about to land. As it came down just across the river, as it came down to the canopy, boom, boom, 2 sonic booms hit you from above. People have warned you about the sonic booms because the rocket has to cross the sound barrier as it re-enters, but no one can like prepare you or brace you for that moment. To this day, every time I remember, I still get goose pimples of foaming on my arms. But ultimately, this was just the start of the experiment. The Dragon then docked with the International Space Station, and the astronaut Scott Tingle diligently put our samples into the Veggie Grow system. Here you can see a lot of people just think astronauts hang out on the side of the space station taking these cool selfies. But there's over like 200 experiments going on at any one time. So like they are really, really busy, and we are super appreciative to have had this opportunity to work with him. We even said thank you on one of the pieces of tinfoil, and he gave us the thumbs up to show that he'd seen it. Really exciting moment. Many years of preparation that took us to get there. One of the great things that Scott did was flew back to the samples each day and took photos of them. And so we had like some time-lapse photography of plants in the corner, and the samples that were in the center, we had photos on day 4 and day 8. So again, there's image analysis and machine learning opportunities with these. These images are in the Open Science Archive, and once the primary PI has published a paper, they'll be publicly available. And hopefully you guys might want to see if you can find some more phenotypes in them. We did so these. This is the time lapse of those plants growing in space. Okay. Hopefully you look at these and you can tell which one's on Earth and which one's in space, right? But what makes you know that one on the left is on Earth? Okay, that's because I labeled it. I know that. But also it's the structure of the root system architecture. You can tell that it's growing down, and the one on the right, they're kind of curving off and going all these funky directions. And so there's biological insights within there that we really wanted to explore at the cellular and genetic level. Maybe some of the engineers and you noticed that the water droplets are forming different too. That's okay. I like having engineers in a lab alongside biologists. I think interdisciplinary projects are much more fun and informative. So because of this particular collaboration, we were collaborating with astronauts, they were able to, and at SpaceX, they were able to send the samples back down to us and not just the photos. And so here you can see Professor Gilroy looking very excited because he has one of these Kennedy fixation tubes that's got some plants in it. They've been fixed and been sent back down to Earth for further analysis. Initially, we used to use the NF Core RNA-seq pipeline as a tool, but we've become enamored with the whole GNAB pipe system and analysis system. So, and essentially, the two pipelines are very similar. But essentially what we did was do the RNA-seq analysis, then put all the data into an interactive relational database known as TOPST. And that's because our primary stakeholders, that the professors weren't experienced coders, and so they wanted a GUI to allow them to compare and contrast between different studies. So if you go to Professor Gilroy's website, the astrobotany.botany website, essentially we've created this and a range of other interactive relational databases using data pulled from the GNAB Open Science API. This allows us to drill down into different subsets of data to create new insights. Okay, cool. So what I'm trying to say there is I had a really exciting experience helping Simon on this experiment. It was awesome. We put the data into GNAB and then we made an interactive data visualization system so we could explore it. But then we needed to find something more from it, some new way of kind of simplifying the insights so we could find some actionable thing from it. And this is when we use a form of machine learning. This is a stress pattern recognition system which would use an autoencoder that took 85 different stress studies from GEO. So these were microarray studies that were publicly available. And then we created what we call a hidden processing layer, as described by our collaborator Fanny et al. This physio space essentially is an autoencoder that allows us to identify 20 distinct types of stress, and then essentially put any study into it and then categorize what type of stress we think that study had. Okay, so what I'm trying to say is we use this system to try and demystify or translate the space-like transcriptome from those plants that were grown in the veggie. It's really important to separate the roots and shoots for this type of analysis because they have quite distinctly different responses to the different factors that cause the plants to grow differently in the spaceflight environment. But essentially, using this autoencoder, we were able to identify the 7 most similar stresses and one which was the most dissimilar. Our hypothesis, as I described earlier, was essentially that the plants would experience a hypoxic stress. And so we were really pleased that this AI system had essentially identified hypoxia as being similar, particularly with the prevalence to roots. I'm just going to focus in on the roots here because it's always nice when you something didn't quite fit your initial hypothesis, but with a bit of deeper diving starts to make sense. So low pH stress wasn't on the radar before this study, but essentially when you think about it, when you have a hypoxic environment, you essentially get lactic acid formation as well as ethyl alcohol and alcoholic fermentation. But essentially what we've come to believe is that the plants in spaceflight are producing lactic acid, and that's essentially becoming a secondary stress associated with long-term hypoxia. So it's a bit like, I don't know how many of you are as unfit as me, but you know when you go running too fast and too far and you get a stitch in the side, that's that lactic acid building up. And essentially the longer the plants experience the spaceflight environment, the greater that lactic acid stress that appears to be, and thus they will start to exhibit physiological changes as a result of that we were able to measure in the root system architecture. So with this with this hypothesis that we're able to draw from the machine learning algorithm and analysis, we then went to the lab and did some more experiments. My colleague on this, Dr. Wong-Yu Choi, went to the Beamer lab, and they did this follow-up study where they used argon gas, this noble gas, to push all of the air out of the chamber, so created a hypoxic chamber. And they used this promiscuous purple pH reporter, which turns yellow when the pH decreases, to essentially show that that in a hypoxic environment, the roots are releasing an acid into the media. So yeah, that was really exciting. My time is still going, hopefully. And then with that, we then had one more step to go. Do I have time? Okay, I've got 5 more minutes. Okay, cool. Okay, so with that information, we then went, well, can we translate this fundamental biology from Arabidopsis thaliana to a crop species? And around this time, Target and the cases, the Center for Advancing Science and Space, offered a project opportunity, a challenge that they call the Cotton Sustainability Challenge. And essentially, we realized we could try and essentially alter the movement of protons to make cotton grow better in stressful environments on Earth and in spaceflight. And I'll describe to you how we did that. So again, I want to list all the team members who took place in this. It takes a whole village to make these types of projects possible. And I really want to thank some of the astronauts for some of the photography and sample harvesting that they did. But why cotton? Well, this is time-lapse photography performed from the International Space Station of the Ariel Sea disappearing as a result of very efficient irrigation systems in the former Soviet Union of Uzbekistan. So essentially, this water that was in this sea is being used in the cotton fields around here. Around this time, there was a technology that would make the crops require less water, and that technology could have been applied. Okay, let's have a look at that technology. Okay, so around that same time as the Ariel Sea was disappearing, Zhang et al. Showed that if you overexpress something called an Arabidopsis vacuola pyrophosphatase, you could make cotton plants that grew bigger and had a higher yield when the fields got drought stressed or when they experienced a saline irrigation. So these are the two major stresses of the terrestrial agro ecosystem, and they're set to increase moving forwards, particularly in the California basin. So essentially, this technology has the capacity to keep crops growing in desertifying environments. It's unfortunate they didn't apply this technology in the fields rather than just irrigating and to the loss of the Ariel Sea. Anyway, so essentially we had this theory that this technology would work in the spaceflight environment because of the protons being produced by the lactic acid. Essentially, rather than forming an acidosis stress, what if the plant was able to pump them into its vacuole and thus use that stress to drive cellular enlargement, cell division, and overall growth? And so that was the hypothesis. AVP overexpression should make the plants be resistant to the stresses of spaceflight and potentially grow bigger. The seeds were sown at the Kennedy Space Center by Dr. Bakshi. This was during the COVID break. COVID era, oh golly. And the astronauts took some photos and sent them back to us. I'm rushing here because I wanted to get to the results and my cat's trying to jump onto my lap. So the photos that they sent back were awesome. We were really excited. One of the things that we realized though was the astronauts, because they're like floating around, they couldn't keep their foot camera perfectly horizontal or parallel to the plane. Fortunately, with Photoshop and other tools, we're able to fix that. But we've since become very passionate about fiduciary markers and integrating computer vision markers into photography. We're always encouraging young scientists to try and do that from the get-go. But essentially, what we were able to do was then start to break down the root system architecture into its primary roots and its lateral roots. Because the hypothesis is that when plants get stressed, they tend to shorten their primary root and send out a more lateral roots as a stress response. And that's what we can see here in the overexpression lines. So this red and green line just here. So this is the total root system length of those GMO lines, the A68 and the D130 overexpression of AVP. And what you can see is their total root system length throughout the 3 to 6 day time point where we had photography was consistently larger than the wild type. These orange arrows, I just wanted to highlight, these are the moments where they sent out extra lateral roots. And so because they were quicker to adapt to the stresses, perhaps due to the way they were had altered proton movement, they were able to get larger root systems and thus get larger shoots. Whoa, okay, so there's a lot going on here. I'm clearly rushing. There's RNA-seq analysis that's ongoing, and I just wish I had the time to try and cram it all in here to tell you some of the fascinating insights that are coming from this. Um, I'm happy to answer any questions if we have time. And more importantly, I really want to encourage you all to come be a member of the Plant Analysis Working Group. Used to be just from GNAB, but now we really are embracing the Open Science Archive and the different types of data that's available. It's more than just RNA-seq that we're interested in. And our goal is to essentially get subject matter experts together to assess these data and then provide feedbacks to the community. And so for you guys as future biologists, future researchers, future bioinformaticians, I would encourage you to try and do time series analysis. So sometimes when we look at some of the old studies, it's just one time point, and that's fine. That makes sense often based on the constraints of space biology. But you guys have the opportunity to change that paradigm and try and push for more time series data. There's a lot of focus on microgravity and radiation, but the atmospheric environment of the International Space Station is dynamic, but it also has very high CO2, and so it's been often overlooked in a lot of classical literature. And so I'd encourage you guys to kind of revisit CO2's role, not just in climate change, but in its effect on human health and built environments. There's lots of people say it can give you headaches, but can it give you something more? I'll let you guys be the judge of that long-winded question. And ultimately, yes, it's been a real pleasure to have the chance to share my research with you, and I'd be happy to answer any questions in this last 1 minute. Over.
That's awesome, Richard. Thank you so much for
Yeah,
your
this is just for
presentation.
fun. Plus a pretty time lapse of cotton plant growing. Because again, I just wanted to highlight that my journey through space biology really did start thanks to time-lapse photography. And okay, it's grown into this crop, cotton, as you just heard, the cotton insights. But this little hero photobombing from the side here is Arabidopsis, that model. And so there's a lot of different model organisms in the Open Science Archive. So I encourage you to find the one that you really have an affinity to and see what journey it takes you off on. Oh, Matthew, I hear you. I see you have your hand up. Go for it.
Yeah, I had like the concept of like plants in like space really is interesting to me. Oh, can you hear me?
Yes.
Hello,
Yeah,
Matthew.
so
If you could, welcome to ask a question.
yeah,
Statistics courses is the most important thing. Actually, one of the funny things that like what that um regression. Yeah, when I calculus. Yeah, one of the funny things I found when I came to US, everyone was talking about calculus and how important calculus is, but statistics is like the underappreciated partner in crime. There, it's so important. And yeah, particularly in this era, this post-truth era, it's really important for you to know how to use statistics so they're not used against you. How about bioinformatics courses? Coursera's got a lot of really good bioinformatics courses. It's a great place uh to search for. Matthew, you got your hand up. You can speak up.
yeah, I was just you were answering someone else's question.
Anybody else? Is anybody here?
Oh, you know what,
Oh, dear.
Richard? I think your
But
your speaker
if you
is
really want to get really good at like the computer sciences and bioinformatics is project-based learning. So it sounds crazy, right? Often you go to classes and they're going to take you through as a pre-made system where wheels really just spin. Um, but fortunately, thanks to James, you guys now all have like real made functioning notebooks, and and thanks to the introduction from Amanda and probably some others, you've now seen the full arsenal that the Open Science Data Archive really has at its disposal. If you guys learn how to navigate that and find something that you're interested in, then restructure those notebooks that James has given you, you guys can do a lot of really amazing research. And hopefully the talk I gave you shows how you can kind of translate some of that into proposals for future flight missions to try and demonstrate like the technologies that you're developing, because that's ultimately the goal, those kind of feedback loops. Oh, okay, so apparently I'm somehow muting. I can't hear him.
yeah.
I can't hear anything. um
Can
Watch
you can you hear us now?
audio settings.
Not yet. Might be like volume.
Hi. Hey, Matthew, looks like your audio isn't switched on though. There's like
Yeah,
that now.
I wasn't
It should
talking
be on.
right. Yeah, can you hear me now?
Oh, but we can't hear you. Yeah, must be
You
something
can't
else.
hear me?
I can hear you. Yeah, we can hear Matthew.
Oh,
So I sorry, audio visual things. In this day and age, we still have these
yeah, I
little
can hear you now,
technology.
James.
Okay, great. Okay, got it. Okay. Thank you so much, Richard. That was really cool. A great presentation. And I think the gist of it, I'm going to continue on, by the way, for everyone, all the students online. I'm going to stay on the call because I know some of you still have some questions about our course. So I just want to hang out and answer those. But I want to just take a moment to thank Richard, Marshall, and Amanda for giving us their presentations and talking about their work and their research. Really fascinating stuff. I know all the students on this call and those who couldn't make it are really jazzed about this stuff. There are a lot of opportunities. We wish to inspire the next generation of scientists, which are you. Or so thank you again for our mentors for all the help you did for the course and for these presentations today. So with that, I'm going to, I guess, maybe stop recording or say, does anyone have any other questions for any of our speakers? Let's see. Okay, Matthew's asking, could you talk a little more about the AWG working group for plant-based biology and the
Oh
opportunities for
yeah.
high schoolers?
Um, so he fell into my trap like a Venus flytraps. I'm gonna drop a link into the forum into a Git book. So it's which kind of describes some of the subgroups like focus areas. But essentially we meet every couple of weeks just to like talk about like the upcoming opportunities for like space biology. So when there's new course proposals and new data sets coming available, new papers, we share them with the community and kind of discuss them. In general, we have people who kind of sign up to give talks, and people that want to create their own like focus group, to come and to like try and get a community of people around to kind of support them with that. So yeah, so you might want to kind of flex some of these new skills that you've got from this particular course. But in order to do that, you need a real project to do. And so coming to these ISS working groups is the perfect place to find someone who has similar interest to you and a similar affinity to it, so you can collaborate on the execution of your new analysis, your next analysis. Yeah.
I should add to that, like, so Richard, do you run that working group, Richard? Is that yours or that's run by someone else?
So it's definitely a team effort. We kind of like run by committee as it were, but I'm the current chair as is. But we do have like annual elections, and so I'm hoping to be deposed soon. Um, But
Got it.
let me drop this link in, and so the more of the story is the fact that you're interested is fantastic, and this particular Git book will essentially outline some of the narratives and provide some links to essentially but yeah, I'll also drop a link to the applying to be an AWG member. So there is like a vetting process to make sure that we don't get vagabonds, hooligans, criminals. I don't know, like someone who disrupt and not be nice. Uh, but I don't think you would fit into that category. But I'll drop that link into becoming an AWG member. But yeah, essentially we then like encourage people to go through few training things. As when we first meet people, there's some GitHub, Gitbook, Gitpod methodologies that we found a lot of biologists don't really know. So as computer programmers, you guys are now really interested in your Python notebooks and getting the data sets to kind of feed into them and get them into your development environment. And so all professors that do these studies don't really know about how to move their data sets to places that are accessible by coders. They tend to get stuck behind like institutional walls. Like and so the more of the story is that we've learned that by trying to get a lot of our stakeholders to kind of understand how to use GitHub, Gitpod, and Gitpod, we can actually help form bridges between some of the old PIs and some of these new programmers that want to create some really interesting new data products. And so again, there's on this link I just shared to the AWG Gitbook, there is a page on like training recommendations. And so they are just some concepts that really stuck out to me from the TOPS open science training. So the NASA TOPS course that was recommended earlier by Amanda. It's brilliant. You should definitely do it. And it gives you a real qualification that goes on your CV, which is great, right? And it's qualification from NASA. That's awesome.
Yeah, exactly. So I would reiterate that. Please do sign up for that TOPS 101 training. It's all online, it's all self-paced. You do get a NASA issued certificate, which is a digital certificate. By the way, for everyone on the call, you will also be getting a digital certificate for having completed this course. But between those, you have 2 certifications on your resume in short order. And the sign up, Sophie, if you scroll back up the meeting chat, there was I think Amanda put it in. I think she dropped it in, or I thought someone dropped it in. If not, Amanda, if you're still on the call, could you drop that link in the chat for how people sign up for the 101 training?
Found it.
Or
Yeah,
anyone.
I'll put that in the chat.
Okay, cool. Thanks. Awesome. And then I should also say about the AWG, so you should absolutely join the AWG. The AWG is a collection of working groups and of analysis working groups. There are multiple of them, and you can belong to multiple of them. So you don't have to just choose one. You can belong to more than one. So for example, I'm working in the AI ML working group. And even in that working group, there are subgroups. So there are different projects going on. So there's a lot of opportunities basically for you. But the first step is to join the AWG, and then you can pick which subgroup which subgroups you wish to belong to. And then you can kind of check it out and see where your interests lie and what projects you might be able to contribute to. So with that, if there are no more questions, I just want to thank you, Amanda, Marshall, and Richard. That was awesome. Really appreciate your time, and your energy and your experience, and to help motivate and inspire these next generation of scientists.
And thank you for your help and your training too. Goodbye.
Cheers. And for the rest of the students who want to stay on, thank you, Amanda. Thank you, Marshall. Thank you, Richard. For the rest of the students who want to stay on, I'm going to stay on now. For anyone who has any questions, and just feel free to write them in the chat, or just unmute yourself and just ask the question or raise your hand or however you want to kind of get in there. But please use this opportunity to ask questions. So let's see here. So someone has a question. I unfortunately have a class that starts at kind of miss. Can I leave some questions here? Working on the predicting lipid density. I'm running this issues doing clustering using DC2 implementation PC. I'm unable to run the following bit of code. I suspect the data set is too small. Yeah, I think we talked about that. Copy of methods implementation, hard code value. Okay, that's right. So I think what might be happening when you do the data filtering is if you filter by differential gene expression analysis, that may be too stringent and you may be down to like 0 genes or just a few genes. And so for anyone who's doing the RNA-seq predicting lipid density using the liver RNA-seq data, you will run into that problem. So don't use the filter by DGA, basically is the short answer for question number 1. Question number 2, copy of method implementations. My GMM has hard code values for metadata. Has hard code values for metadata. Okay, so you can change that. So you'd have to get into the code for right. So go to my GMM, which is in the methods notebook, right? And just change whatever is hard coded in there. Change it to match up with the data sets that belong to the liver data. Number 3, while plotting the heat map, the Grapherd data frame shape is 1 by 41 columns, and the heat map cannot be plotted. clusters do not gene interfere. That's I think it's the same problem. So I think it's just a continuation. Number three is a continuation of having filtered by differential gene expression analysis. I think that filtered down to 0 genes basically, and so all the subsequent things analyses won't work. So just don't. So take out. So just redo that notebook, but take out or comment out or skip the filter by DGEA step. Don't filter by differential gene expression analysis because it filters everything out. Last question, once the RNA-seq data sets are merged, the data set has NANs looking like random forces network with NANs. So again, you might, again, I don't know about this, but again, if you have NANs, you can take out NANs, right? That's one of the filtering techniques. Um, um, so you can remove NANs, and hopefully this is not still a continuation of that problem with filtering by DGEA. So that one you would potentially have NANs in your data set. So, but you can filter out NANs. There's something in the My Methods notebook that has filter out NANs. Okay, Sophie, you have a question. Is it all right if we submit the capstone evaluation writing project? My partner and I have been going through coding issues, but I can still do the other capstone project and email you with questions, even though it's past the due date. Submit the capstone evaluation writing project. Absolutely. Evaluation. So that's fine. Yeah. So I will accept any and all of those, including something that you chose. I think I had that as an option, but you can certainly pick any one of those. Are all equally valid. They don't all take the same amount of time or effort. I can say that. But that's not really the point. The point is to explore something that was meaningful or interesting to you. So if at this point you're not able to kind of finish the project you wanted to finish, and then you just want to do the evaluation, that would be great. Do note that I will be giving you all a survey. So you will all have a mandatory opportunity to provide feedback about the class. I haven't issued that link yet, but I will be sending out that survey. So, but I am accepting the evaluation writing project as a capstone project. That's perfectly fine, Sophie. Other questions, please feel free to raise your hand or just unmute yourself or write it in the chat. I'm happy to take your questions.
Had one question.
Sure.
Yeah, so I am working on like my own project. I think I got this approved by you, but it's it's based on like data I found on the AWG like OSDR open science website
Okay.
on it's like plant growth based upon in 0G based upon different percentages of like minerals. And I've been trying to find like use linear and multiple regression to determine like to predict the height and other aspects of the plant after a certain amount of days based upon like a input of um, like components of the soil. I just
Okay.
had some like questions regarding like would there be able like is there a way to like graph my data? Like I'm just unsure how to do that.
Right. So basically, if you think about it, if you like, in terms of regression, and it sounds like you're doing regression, right? You're predicting some continuous value based on other continuous value features.
Yeah.
Is that right? Yeah.
Mm-hmm.
So if you think about it, the typical y equals mx plus b line, equation for a line, the slope intercept form of a line allows you to graph something in the plane. And by the plane, I mean in two space. You have two dimensions. You have an x and a y, and you're expressing or graphing y as a function of x, where x is a single it's a univariate function. If you have multiple features, like, you have, I don't know what the features are of your data set, but suppose you have like amount, temperature and pressure and density of soil, or some amount of carbon in soil, or amount of some, suppose you have 3 features or 4 features or 5 features that are predictive of your plant height, or whatever your phenotype is that you're observing, your response. You won't be able to graph that in 2 space, because you have like, it's a 5 space or 6 space, right? So you could, if you wanted to graph something, you can graph something as a function of one of those variables. So you can do one variable at a time. So you can graph, I think you said plant height. Let's just say it is plant height. I don't know what it is, but let's say it's plant height is Y. And you can graph that, and that Y, and then only include on the x-axis, like, each one separately. So if you had 5 explanatory variables or features, like, amount of water, amount of sunlight, pressure, temperature, whatever those 5 things are, you can create 5 graphs. And then on the x-axis would be each feature one by one. And so you can see how Y is responding to each feature one by one, but you wouldn't be able to put it all in one graph, because we can't visualize stuff in more than 3 dimensions.
Okay, thank you. That makes sense. Yeah. So like I have multiple inputs and a few outputs, which is like plant height, leaf area, and one more, and plant mass. So I guess it wouldn't be able to do that.
Yeah, but you can, like I said, you can create separate graphs, not put everything in one graph, but create separate graphs where you have your Y as a function of each one of those separately. And then you can see you can see the relationship between Y and X separately, in those different graphs.
Okay, I see. I'm trying to, yeah, so I'm trying to also find like an ideal like soil composition, which is like nitrogen, carbon, calcium, like atomic components. Um, and those are my like X values.
I
Like, is
see.
it possible to find an ideal X, like amount of X values per, like, to get the maximum Y values, or a single Y value even?
Yeah, that's a good question. So there are ways to do that. One thing you can do is just try, let's say you had three X variables. So how many combinations of those 3X variables do you have? It's basically um I don't know if you know this syntax, but there's like a combination of 3 choose 1 plus C 3 choose 2 plus C 3 choose 3. So you could um so this is using this combination syntax. So there's like, how many ways are pick. So you can pick one at a time and see what your performance is. Then you can try any 2 of those. So like X1 and X2, X1 and X3, and X2 and X3, right? Those are the different combinations on 2 variables. And then you can try using all 3. So there will be these are all the different ways of combining, using different numbers of those 3 variables. So you'd have to, so this is 3, there's 3 of these, there's 3 of these, and there's 1 of these. So I think there's 7 total combinations you'd have to look at, and then you can see which one optimizes your response variable. So
Oh, I
you
see.
just do it exhaustively, right? Just look at all possible combinations, right? Just each one by themselves. That's this guy right there. All possible pairs of X values, and then all X values together. So just look at them all, like, just explore, it's exhaustive, and then just, look at all of them and which one gives you the best performance.
Okay, so there's like no, it's not like it can, it's not like it creates a multi-dimensional graph and then can select the exact variable where like the absolute maximum is, right?
I don't see how it can. Yeah, I'm not sure. I don't see, I'm not sure. I can't personally, maybe there is, but I can't think of a way to do that. Like this is how my brain would work anyways. Just try all possible combinations and then see which one gives you
Yeah.
the optimum.
Okay, I understand. It's like a lot of variables. There's like over 10 of them. But
Yeah, you have 10, yeah. So you would have a lot there. Now you can do feature selection before maybe not all 10. So the other thing you can do is see of your 10, which ones are most correlated to your target, right? So if something is not correlated to your target, you can just throw it out, right? And so you can just calculate the correlation coefficient, right, between each of your 10 variables and the target. And and then just have some threshold cutoff. Like if it's 0.5 or below, throw it out. If it's greater than 0.5, then you keep it and then you do this all possible combinations. Because if something's not correlated to a target, it's not going to be able to predict it. So you have to, if it's, so you start with correlate. That's one way to filter out meaningless features. Is if it's not correlated, it's not gonna be able to predict it.
okay, thank you. That makes sense. Also, is it like one last quick question? Is it normal for like there to be a large margin of error, um like the when I tested one of them, one of the points, it came out to be like 2.98 is the actual, but the one that was like suggested by the AI or the machine learning algorithm was like 2.17. Is that large of a like error normal?
Yeah. So you will have higher error in your model, basically, like whether it's mean squared error or mean absolute error or R squared, depending on how you're measuring your performance of your regression. But if you have a lot of variance in your data, right, then you will have a lot of error, a higher error. And if you only have a few samples also, you'll have higher error. And I'm guessing both are true. You probably have high variance and you probably only have a few samples. And so, yes, having high error does make sense in this
Yeah, I
case.
only have like 20 samples, so I
Yeah,
guess that'd
yeah,
be correct.
yeah.
Okay, thank you. That's all.
Sure.
That was really helpful.
Oh, good.
And I could
Good.
just submit it anywhere on I could submit on any where do I submit it again? Is
For
it like a
your,
specific because
do you
there's
mean for,
multiple capstone projects I can submit it under.
oh, I see. So, well, you mean which one does it apply most to? Is that what
Yeah,
you're
which
saying?
one do I choose? Because yeah, I did something a little bit
Oh,
different.
it won't matter which one you pick.
Okay,
It
thank you.
won't, yeah, as long as I get it in under one of those, it doesn't matter which one.
Okay, thank you.
I think the next one, let's see, I'm just looking at is it all right for some writing project? Got that. Rowan, did you solve your problem for the research paper? How, okay.
I see.
Shri,
Yeah, I actually had a question regarding my project. So,
okay,
um, yeah, I've been emailing you about like my progress so far, but
yeah.
I just wanted to clarify my understanding of like causal inference and the effect that invariance has on it. So, from my understanding, the point of I guess causal inference as a field is to be able to develop inferences between like the predictors in a model and the results it achieves, right?
Right. So the idea is between invariance and causal invariance and causal uh, sorry,
Causality.
invariance and causal inference. These are a mouthful. The idea behind them is um, if something is truly causal of something, then it won't matter the environment that you're in. Like, for example, we
Right.
know now that smoking causes cancer, right? And it doesn't really matter if if you also drink coffee or if you drink smoke, and I should say lung cancer. No, no, it doesn't matter if you like, if you're if you're 6'7" or you're 5'3" your height doesn't really have much of an impact on that. So regardless of your height, regardless of your weight, regardless of your gender, regardless of your socioeconomic background, regardless of your diet, regardless of all this stuff. So invariant of all the other things that might be part of your background, smoking is causal of cancer, of lung cancer. So that's generally the idea. It's not, in other words, it's not, there are no, what do they call those? There are
Confounding
no
variables.
confounding, thank you. That's exactly the way I was looking for. There are no confounding variables there that, smoking causes something else that causes cancer, or something causes you to smoke that causes cancer. It's nothing like that. It's like, another way of looking at it is what causes you to want to smoke? Well, it doesn't really matter, right? It's the smoke, like, well, I get stressed out, and then when I get stressed out, I smoke, and then the smoking gets, so getting stressed out causes cancer, or that's not what we say, right? So it's invariant of whatever the those confounding factors, or colliding. That's another type of factor. It's independent or invariant of that. And so that's the idea behind invariance. And so you basically have to capture, if you want to use invariance, then you need other factors. You need other environments, like you need other information, to be able to capture that. And so Rowan, I know you're looking at data augmentation. So those augmentations themselves become environments for invariance. So in other words, it doesn't matter if we log transform this data. It doesn't matter if we take the square root of this data. It doesn't matter if we take the centered log ratio of the data, it doesn't matter if we normalize data. No matter what we do to the data, these are the predictors that are most predictive of the response. So you can use data augmentation as in interventions or environments in your causal inference when you're using invariance. So they kind of go hand in hand, and it's a 1 + 1 = 3 situation, because number one, using those data transformations to augment your data gets you more data, which is what you need, right, in machine learning. So that's good that way, but it also gives you just built-in environments for invariance, which is what you need for if you're going to exploit invariance for causal inference. So those two things go well hand in hand. In fact, I'm just submitted a paper to get published. Hopefully, it gets published, exploiting this very relationship between causal inference using invariance and data transformations for augmentation.
Right, yeah, I was reading through your paper regarding like CRISP and the machine learning ensemble that like selects the most
That's right. That's the
correlated
one.
feature. Right, yeah.
That's
And
the paper.
it's really interesting work. So in my projects, I'm using random forest, and I understand that it's not typically a casual predictor, or sorry, causal predictor. But in implementing invariance, does that allow it to,
Yeah.
or does that allow us
Yeah,
to like
that's exactly right. So the way, if you
inference?
look, if you look at the paper I posted in here, their implementation uses something called empirical risk minimization, but it doesn't, it doesn't matter. It's you can apply the concept of invariance to whatever algorithm you want to. There,
I see.
it's all the invariance thing it's a matter of splitting up the training and the testing data and validation data if you're doing validation. But let's just, to make it simple, if training and testing, what you do is you train in one environment and you test in all the other environments, right? So you train your model in one environment, so you adjust all your adjust all your parameters of your model in using data from one environment, say the log transformation, and then you test it against the data from all the other environments, like the centered log ratio and the normalized data and the square root data and all that stuff. And then you continue on and you kind of cycle through each of the transformations, train in one, test in all the others. And so you can certainly do that with random forest. And I'd be curious to see what you come up with, because that's something in the back of my mind I've always wanted to do but never did, was because random forest is a great, great, well understood algorithm, and I think it would be really cool. And even if it's not for causal inference, anything that's it will just produce more robust correlations, right?
Right.
So, it doesn't have to be for causal inference. It can just be, you can just say these are more robustly correlated. These features that my algorithm found are more robustly correlated with the targets because it's invariant of, it doesn't matter what background it is. It doesn't matter which, environment I test this in. It's true, invariant of that. And so that's a more robust correlation. Doesn't have to even be, you don't have to even use the word causal.
Right. So, do you mind if I share my screen real quickly?
Sure.
Uh, okay. I'm going to share my VS Code screen. So, can you see this on?
Yes.
Okay. So, for my model results, I already have pretty strong results for like my two target variables, which is the Jalal cells and the TH positive cells. So for the inference part of this, would you recommend that I simply use the features that these models selected, or should I find like the most optimal features using all of the models?
So that's a good question. So you can either using all of the models is kind of like an ensemble approach.
Mhm.
And so if you use all the models, that there's, um, and then just see basically where do they agree, right? So you have 5 models, say, and they have each come up with a list of 10 features, say. You can just find the intersection of those 5 lists of 10 features, and then you can call that that's an approach. It's called an ensemble
Mhm.
approach. So that's a way to go. The other way to go is to say instead, just find like the ones that are most predictive, period, right? And, they don't have to intersect. They could be instead of doing an intersection, you can then do a union of the most predictive features from each model.
Right, I see. So like the highest frequency of predictors within each one.
Right.
Okay. And in my paper, given that I don't necessarily have a lot of time to I guess add more stuff to this, I was just wondering if it's okay just to use the features from one model.
Yeah,
Yeah,
absolutely.
since like the results from this one are already pretty high, and
Yeah.
yeah, it seems that like random forest just seems to dominate in this data set.
Yeah, random forest is it's a well liked, well used algorithm for this purpose because it doesn't depend on like it partitions the data, the feature space, in a way that doesn't depend on it can tolerate outliers, it can tolerate different scales of data. So
Mhm.
it's a really well used algorithm. So yeah, you can just use that one. That's fine.
Yeah, okay. And I actually didn't know to like when I augmented the data, I didn't know to use each like transformation as a different environment. So like so far, I've just been using the environments as like Earth or
Yeah,
microgravity or artificial gravity.
right.
And so going forward, like each of these or each of the physical environments will have another environment for each of the transformations, right?
Correct. So
Okay.
you would compose the environment. So you basically have Earth colon log, Earth colon square
Mhm.
root, Earth colon normalize, Earth colon box cox, right? You would
Right.
you would do a cross product of those. So you would basically like that.
Right, okay. And so I was wondering like the what the interpretability of that would be. So if the highest performing model was tested in Earth, or I mean trained in Earth, but was tested in microgravity and was the most highly correlated, what would that mean?
I don't know. I think that would be very like data set dependent, in terms of the interpretability. I think like for now, I think like you can just kind of take it at its word and just say, you know, which features did it come up with, period. But if you wanted to kind of explore underneath the hood and see why that's happening, I think you'd have to look mathematically at those data sets and what's the relationship between the model that's produced on Earth data and tested on microgravity. You'd have to kind of do some sort of mathematical analysis to see. My guess is that those probably have the highest similarity are
I
the,
see.
you know, is my guess. But like I said, you'd have to kind of analyze that on a case by case.
Yeah, it's interesting because the mission where this study took place, they try to reproduce Earth's gravity using a centrifuge and test if like CMS function changed in space. So I guess it makes sense why uh, like the artificial gravity and Earth's gravity are similar in results.
Yeah, that makes sense. Yeah, from that perspective, that would make sense. Yeah.
Mhm. And so within my paper, to discuss the model results, um, I believe you also did this in your paper as well, but I was just, or I would first discuss like which models were highly or the best performing, right? And why they were based on like their architecture and I guess the data set.
Yeah. Yep. That makes sense. So you're writing that's what you're gonna submit as your capstone? Is
Yes,
it
I
is it
actually,
okay.
yeah, I can send my paper in the chat again.
Okay.
Yeah, so far I've just been working on like the introduction from like the paper standpoint, and then I've been doing the coding on the side. But to discuss like the model and I guess the results from it, um, I do select about 2,000 genes, and I do want to talk about like which genes were selected for this and how um and like why they were. So how would I do so? Like of the 2,000 genes that were selected, which ones would I guess be most optimal?
Right. So first you have to say how in your paper, what you all you need to do is say how you got down to 2,000 genes. Like so what were your filter criteria to get down to 2,000 genes? And then of that list, I'm guessing you're going to have some subset of 10, 5 or 10 or 3 or 15 or something genes that your models found, right? That they identified as most predictive of your response variable.
Well, actually, sorry, before you continue, the 2,000 genes I used were selected using feature selection algorithms such as like cross validation, RFE, and things like that.
I see. So yeah, just mention that. You'll need to say in your that's just what you need to say in your paper is just say
Okay.
that. Yeah.
Okay.
Yeah.
So yeah, knowing that, do I have to discuss uh like what genes were selected and why they were?
You mean like which genes? So first the selector selected 2,000. So you just say in a sentence or two, like we use these things to select, you know, do feature selection. But then right, and then you will just say for now my models found this, you know, these 10 genes or how many ever genes from the 2,000, it found 10, say. I'm just going to make up that number. You just have to say you do have to describe which genes it found and how it found them. So it found it using the weighted coefficients. We just took the absolute values of the coefficients and picked the biggest ones, or you did Shapley feature importance or whatever you did to do feature. I know random forest has its own built-in feature importance, so just say that. So then just say we used random forest's built-in feature importance to select the top 10 or 15 or how many ever you select.
Mhm, I see. And like from like a biological standpoint, each of these genes or yeah, each of these genes like express a different part of the CNS. So do I have to talk about that as well? Like from like the metadata descriptions of it.
You might, I mean, that's, you know, so you might dig a little, so right now, so the interesting thing about the research we do in this space is, you know, we can treat these like black boxes, right? We can just say like, these are just features, right? And this is the response, and these are the features, and not have try to have any understanding. But then if you want to dig a little deeper, it's not just machine learning that we're doing. We're also doing, we're analyzing biological data. And so then the 10 or 15 features that your model found, right, as most predictive of whatever your phenotype was, you would want to know what those genes are, what pathways those genes enrich, using pathway enrichment analysis, and what gene sets do they belong to using gene set enrichment analysis, and what proteins do they encode, or are they protein coding genes? Like what are those genes? What are they involved in? Why would that, does it make sense that these genes are predictive of this response? That's ultimately the goal of the research, right? We use ML to identify the genes, but then we have to use biological reasoning and logic to see if that makes sense, right? Does it make sense that, that these genes would be implicated in this response?
Right. So on that note, I wanted to clarify like what role I guess pathway analysis would be in this, and like from what you just said, it would just be to uh, help us understand what genes are used, right? And
Right.
why
So
they.
yeah, so you'll get a list of, say, 10 genes. I'll just use that number again. So say you have your models came up with 10 genes that are most predictive of your phenotype, of your response. Then you take those 10 genes and you go to a tool like Shiny Go, which I've used in the class, which is a pathway enrichment analysis. We also have pathway enrichment in Python, but you can go here and you can just plug in your genes, literally just copy paste the list of your genes, paste them into that tool, and it'll give you a list of pathways that are enriched by those genes. And then you can say, "Hey, are those pathways related to my response, right? To my phenotype, right? And if they are, then it's a form of validation, and it's also a form of understanding, like what is it about these genes that is causing this phenotype? So the pathway analysis will help you with that. And so will gene set enrichment, right? So if you go to MSigDB, the same thing. You just copy paste a list of those 10 genes, and you just copy paste them into this tool, and the tool will find the gene sets that are enriched by those genes. So that's the next steps. That's where you start the bio. And then the other thing is you just Google those genes, right? Just look them
Right.
up. What do those genes do? Does it make sense that those genes would be implicated in this phenotype that I'm observing? So it's, there's a lot going on at that point. So, really, if you wanted to really validate your findings here, what you'd really have to do is do some sort of in vitro or in vivo test, which we're not going to be able to do here, right? We're just not, that's not the scope or spirit of what we're doing here in this class. But in scientific research, the next step after identifying a putative set of genes predictive of such and such, you'd want to do an in vivo or in vitro test of that. Is this how it really plays out? Either do knockout experiments or knockdown experiments, or some sort of like, gain of function or loss of function in vitro. Is Do we observe this phenotype in the real world, like in biology, um, through the manipulation of this gene expression? So
I see.
so that's really ultimately what, if someone wanted to find out and maybe make a drug, right? Like, hey, I'm in a, I want to, I'm in a pharmacological setting, and I want to see if I can actually create a drug that inhibits the expression of this gene. Does that have this impact on this phenotype, right? So that's where you then start to do these things, like, in real biological experiments. That's kind of would be the next step.
Right.
So,
I see.
so what we do here is just we create the putative list of genes. These are genes of interest, right? So for those of you out there in the biological world who want to experiment with these, here's a lot of evidence that these genes are causal or at least robustly correlated to this response. Then you leave it up to the biological experimenters to actually, if they want to, then test that out.
Right. Yeah, I'm looking at your paper right now, the one with CRISP, and yeah, it seems that you just listed the genes and their functions from like the gene databases online.
Right, right. So I
Okay.
just kind of say, yeah. And I say, it makes sense that these genes would be predictive or causal of a high lipid density, right? Because they all function in that capacity. So
Mhm.
it's just evidence. But the best evidence is what's called they call it a what do they call this? Sorry. It's a what is this? Why am I losing the term right now? It's a controlled experiment, but it's not a controlled experiment. It's a randomized controlled trial. That's what it is. Randomized controlled trial, or RCT. So that's kind of the gold standard in science,
Mhm.
where you randomly assign samples to experiment group or control group, and you run a controlled experiment, right, to see it, to play this out. So, and the only difference between the control group and the experiment group is this thing, right? This gene, right? And it's, either knock it out or knock it down or whatever, however you manipulate it. And then you see, does this lend itself to that phenotype, right? Does it,
Mhm.
and that's, so that's the gold standard. But there's a lot of steps before you get to that, right? So before you get to that randomized control trial, which is very expensive and time consuming, what are the genes we want to test? Right. And so that's what we can do with AI and ML is we can come up with a list of genes that is putatively causal or putatively meaning possibly
Right.
causal of this phenotype. And that's what we do. And we do our best as, in silico, to predict what would happen in vivo or in vitro.
Right. So yeah, this is all really cool to me. I took AP statistics last year, and so I learned a lot about like all the statistical terms you were talking about, and it's just really cool to see it applied to a field like space biology, and everything just is just coming together with like machine learning too. And last question for you is regarding the paper. Uh, did you hear back from anybody regarding the?
No, I didn't. So I did. So I made a call or sent an email a long time ago. They responded like way back when you asked. And then I went on vacation. And then when I got back, I just this earlier this week, I reached out and haven't heard back yet. So there's a bit of phone tag going on. So just sit tight. I would like to see if we can get this published in that journal, and I'll make this option available to other students. So as soon as I find out from this person, I will let you and everyone else know so that we have somewhere where our papers can land. But right now, I haven't heard back yet.
That would be really cool. I am
Yeah,
applying to college this fall, so like having a paper already published in like an NASA journal would be cool. Because I do know that other journals take like months and months to finish.
it can. That's right. So the other thing you can do right away is you can publish it to the bioarchive. Are you familiar with that?
No, no.
Okay, so bioarchive. It's basically a place where you put preprints. So this is you should register for an account here, and then you can publish this here. It's not peer reviewed, but basically you have a copy of it out there on the internet that you can point people to. And then it's a way of preventing your research from getting scooped also. So
Mhm.
scooped meaning someone steals your research idea and gets it published before you do. So this is a way to kind of say, hey, I was here first, right? I put my paper up here. And so it's something you might consider doing as this bioarchive. That's kind of what I've done as a student. They encouraged us to get it as soon as you're done with the paper, get it there and then submit it to your journal.
Right. And I actually have one more question. I know I said my last one was the last, but I just thought of this. Regarding the AWGs, in like the AI ML AWG, you recommended that I join the casual inference one, but it seems that that one is already um like working on a specific project, like regarding casual inference. Like as part of the casual inference subgroup, would I be able to like start another project within that?
Yeah, absolutely. You could. You could absolutely start another project. So yeah, we're working on something right now, but absolutely there's a lot of members, and so you just basically present your ideas and see who else is interested. And then you can, so it's never too late. It's one of those things, it's never too late or too soon. Everyone joins when they join, and you can either join an existing project or you can create your own. So it's just a matter of kind of getting other people to kind of, you present your work and your ideas and you kind of see what other people might share your research interest.
Yeah.
So with that, I'm sorry, I gotta take other people's questions because I'm actually running out of time myself, but thank you for your questions. Um, I'm just going up here. So Bella, you have a question for the research paper capstone. How many images figures should we include? Should we write captions, describe the pictures as well? So yes, you should. So usually, it's something like, like 3 to 5 figures is appropriate, something like that. And some journals will actually have hard limits on it, but 3 to 5 is appropriate. And yes, do include figure captions. I'll always do that to explain what's happening in the figure. Okay, and then will the research team form be sent out soon? Okay, so thank you for that question. Yes, I will. I'm just making a note of it too on the side. The paper research pub team. Okay. Yeah. So I will do that soon. And I will send that out to everyone who's then everyone who's interested can reply back. Would we be able to, some that we're speaking today if we have questions? So I think Matthew already answered that, but yes, please do reach out to them. They put their emails in this chat, so you can see. Let's see, if you scroll? If you scroll up in the chat, you'll find Amanda's and Marshall's and Richard's email addresses. Sorry, I'm not seeing them right now, but I know they posted them in the chat. Let's see here. Where was that? Oh, it should have been towards the end. I know I saw them post them in there. Why am I going too fast? No, maybe it was at the end of Marshall's presentation. There we go. So there's Marshall's, there's Amanda's, and then this is how you can reach Richard. They're all from 2:04 p.m. You can see all three of them back to back to back. They put their how to contact them. So please feel free to reach out to them. Yeah. All right. So I guess with that, that's it. So thanks everyone for attending and for all your questions. And good luck finishing up your capstone projects and all your notebooks. Feel free to continue with your sending questions to the discussion. I'm going to leave the Canvas open for some time. So if you continue to have questions, whatever, after class is over, feel free to use the discussion board there. You can also feel free to email me directly and look for survey links. I'm going to send out a survey. I will be issuing certificates. Please do take that TOPS 101 training that was referenced earlier. That's another certificate that you can add to your resume, issued officially from NASA. This is not, by the way, this is not an official NASA class. This is funding that was provided by NASA to create the class that we took. So I'll say as much in your certificate, but it's not like an official NASA certificate because they don't issue certificates like that. And I will be posting information about those of you who want to participate in writing that paper and collaborating to write that paper on the research that we did. So with that, I'm going to close out. Thank you, everyone, and have a good rest of your day.
Thank you, Dr. Castelletto.
You're
Thank
welcome.
you.
Thank you.
Thank you.
Thank you.